Hello everyone, my name is Zhang. I'm from the University of Electronics Science and Technology of China. I'm very honored to talk at the tutorial Tractometry Peering into the White Matter. So my topic today is about how to quantify fiber bundles using diffusion MRI tractography. So first of all, I would like to apologize for not being able to present in person due to a conflict of the time schedule. So I'm doing this video presentation here. At the end, I will need my contact information, and I'm happy to have further discussions related to the talk today. I have nothing to declare. So speaking of how to quantify fiber bundles using tractography, it is still a challenging and open question in the field, given the fact that there are many options to choose from, and each has its own pros and cons. So in general, when we want to perform a study related to the fiber bundles, there are several major steps, including how to segment tractography into fiber bundles, how to extract quantitative measures from the bundles, and also how to analyze these measures so it can be used to help us understand the brain in health and disease. So working with several experts in the field, our group published a review paper about 2 years ago on how to use tractography to perform quantitative mapping of the brain structural connectivity, which also includes the content that I'm going to present today. So if anyone is interested, please also consider taking a look at this article for a broader picture. So this slide gives the agenda of my talk today, including how to perform tractography segmentation to identify fiber bundles of interest, and then what quantitative measures can be computed from the fiber bundles, and following by how to perform data analysis using the computed measures, and finally what we can do in neuroimaging-based applications using the bundle quantification. I will focus mostly on the second and the third parts and briefly talk about the first and the last one for completeness because they will be covered by other talks in this tutorial. First, so the goal of tractography segmentation is to identify white matter fiber bundles that are meaningful for quantification of the brain structural connectivity. And in general, tractography segmentation methods can be grouped into two categories related to the bundle-specific analysis, that is usually hypothesis-driven and studies certain anatomical fiber pathways. And the other is connectome-based analysis, that is more data-driven and studies the whole brain structural connectivity between different brain regions across the entire brain. So the first category of tractography segmentation methods identifying the anatomical fiber bundles and label them with traditional names. For example, on the left, you can see a brain dissection image showing the distribution of the actual white matter fiber pathways. And what you can see in the red circle is called uncinate fasciculus, that is related to the human language. And on the right, you can see what we can get from tractography segmentation, showing a very good correspondence to the brain dissection. That's one of the major reasons that we want to use diffusion MR tractography to study the brain. And currently, there are many ways to perform the anatomically named fiber bundle identification, which has been covered by Professor Anastasia Yandiki's talk. So please refer to her talk for more details. And the second category of methods parcelates or subdivide the entire white matter into many parcels. So one widely used method is called the cortical parcellation-based, as shown in the left figure here. So it works from a gray matter-centric perspective by parcellating the tractography according to a cortical gray matter parcellation, and it extracts streamlines that connects between pairs of ROIs. So on the left, you can see an example cortical parcellation derived from a free surface software and the fiber bundles connecting the precentral gyrus and the brainstem. And another way to parcellate the whole brain tractography is called fiber classing methods that works from a white matter-centric perspective. So these methods group the streamlines according to their geometric trajectories and describing the structural connectivity according to the white matter anatomy. And on the right figure, you can see a whole brain tractography parcellation image into many small fiber clusters, where each cluster has a unique color, and the streamlines within each cluster has very similar geometric shapes. Of course, there are also many other methods that leverage both cortical parcellation and streamline trajectory information to enable the joint analysis for improved segmentation. And we have one paper published in Neuroimage last year within this direction. So if anyone is interested in this kind of research, please also take a look at our deep fiber classing method. Then after performing segmentation, the next step is to compute quantitative measures that are useful to assess the structural connectivity of the fiber bundles. And in the next following slides, I will introduce 3 major types of measures that are widely used, including the connectivity strength, tissue microstructure, and the shape measures. So for connectivity strength, so the number of streamlines, that is NOS, is one widely used measure. So as shown in this figure, with NOS measured from each fiber bundle obtained from whole brain tractographic segmentation, we can build a connectivity matrix that allows modeling of the entire brain as a graph. So this enables data-driven analysis to study the structural connectivity of the entire brain. And this is widely used in the literature to study the brain's structural connectome that I will introduce more shortly in the later of my talk. Please be noted that because of the difference in the resolution between diffusion MR signals and the actual axon dimensions, so studies have emphasized that the NOS is not a true quantitative measure of the connectivity strength. That's why here the strength is quoted. But recently, more advanced work has been done to provide alternative measures of the connectivity that can better estimate the underlying connection, for example, using filtering methods or many other techniques. And the second category of quantitative measures is called bundle tissue microstructure. So this is done by mapping microstructure measures at each location along the fiber tract, as shown in the top figure here. And the most widely used are the measures computed from a diffusion tensor model, such as the FA that quantifies the diffusion anisotropy of the water molecules, and the MD that quantifies the overall magnitude of water diffusivity. Of course, there are also many other modeling methods, such as the free water, NODI, diffusion kurtosis, which can all be used for computing bundle tissue microstructure. And in addition to the diffusion MR data itself, we can also use other quantitative imaging modalities, such as the myelin imaging for analysis that are beyond the diffusion MR. Then after choosing a quantitative measure of interest, we can quantify fiber bundle at many scales, shown in the bottom figure here. One way is to use a scalar value to summarize the entire tract, and the mean measure within the fiber bundle is the most widely used. Other summary statistics, such as the median, the maximum, the minimum, have also been used before. And the second category is to measure the distribution of the microstructure measures along the fiber pathway. And this approach requires the definition of a coordinate system or assembling framework to define how to sample the microstructure data of interest, as seen in this figure. So unlike using one scalar summary statistics, we can see that a long bundle representation can provide more information specific to the local fiber bundle region. This is also the key representation of the tractometry techniques that will be introduced later. And the third category of the quantitative measure is the shape feature. Then compared to the aforementioned measures, the shape features have been relatively less studied, but it has gained more and more attention in the field. So early work has attempted to measure the trajectory or the overall shape, surface shape of the bundle of interest. But more recently, studies have tried to extract many other features, such as the diameter, the span, the radius, to quantify the fiber bundle shape. And our work published this year investigate a point-wise representation of the fiber bundle that enables the study of a detailed spatial extension or determination of each anatomical fiber bundle. For example, we show that this approach can help us differing terminations of the articulus language tract across subjects and identify the fiber regions that are predictive of individual language performance. Then, once a quantitative measure has been extracted, several options exist for bundle analysis, which can be hypothesis-driven using statistical analysis or data-driven using machine learning. So these methods may rely on summary statistics per tract or data along the fiber bundle using tractometry or using a connectome-based analysis. One widely used analysis is based on a hypothesis-driven strategy to assess if there are statistical differences of fiber bundle of interest between different groups, for example, between health and disease or between different subtypes of disease. And usually given a quantitative measure, a summary statistic is extracted from the bundles in all subjects under study. Then the level of group differences is computed using group-wise comparison methods, such as the student's t-test and the ANOVA. And another hypothesis-driven analysis uses a regression model to assess the correlation of bundle quantitative measures and to a behavioral or a disease symptom score. Such analyses have been applied to study how fiber bundles are affected in individuals with different disease severities, how tracts relate to cognitive functions, or how tracts are developing during the neurodevelopment and aging. And these analysis methods will generally give a p-value. So if the p-value is lower than 0.05, it means these analyses have statistically meaningful results. So one important point to note here that the correction for multiple comparisons is needed if there are multiple tracts or multiple quantitative measures analyzed. And commonly used multiple comparison correction methods, including the FDR and the Barfoni method. Another bundle-specific analysis strategy uses machine learning to perform tasks such as disease classification and prediction. So in machine learning analysis, the quantitative measures computed from individual fiber bundles are treated as feature descriptors, which are input to a machine learning algorithm, for example, a support vector machine or a convolutional neural network. This can train a model from a set of training samples, and then the trained model can be applied to new samples to predict a class label. Unlike statistical analysis that is usually aims to find white matter structures with statistical group differences, the machine learning-based methods aim to offer predictive relevance. And one benefit of using machine learning is that it can be easily applied to multimodal data and can leverage the information provided from a large-scale dataset. Then instead of using a scalar value to summarize the entire fiber bundle, many methods perform a long-track analysis to investigate the distribution of microstructure measures along the fiber pathway. This is usually called the tractometry or the tract-based morphometry methods. And a long-track analysis enables the study of the local regions along the fiber tract. And quantitative measures computed from individual fiber bundles can also be used to construct a structural connectivity matrix to map the whole brain structural network between all pairs of gray matter regions. And this approach is motivated by a general shift of the focus away from the role of isolated regions towards understanding the brain as a network system. And in general, a structural connectivity matrix defines a network or a graph of the brain, where the nodes are the gray matter regions and the edges represent the white matter connections between these cortical regions. And the disciplines of the graph theory or the network science provide a framework to quantify the network probabilities of the brain connectivity and investigate the whole brain models for structural varying. And in the last 2 decades, diffusion MRI tractography has become a key technique for study of brain's white matter connections in a wide range of applications. This includes the brain development, aging, neurological disorders, mental disorders, neurosurgeries, and etc. Here I would like to refer to the following talks for details about the application of different tasks. So here is almost the end of my presentation, and I would like to give a brief summary. So there are many ways to quantify fiber bundles using diffusion MRI tractography, and popularly used quantitative measures including the connectivity strength, the microstructure measures, and the shape measures. And regarding analyzing the measures computed from the fiber bundles, it can be done either using statistical or machine learning analysis methods, which can enable the study of the brain-white matter connections in health and disease. And machine learning and AI approaches can further improve the data processing and analysis, and further improve our understanding of the brain's white matter fiber bundles. And finally, as a note, I would like to say, where there have been considerable technical advancement and a broad of applications, there still remains no consensus about the best methodology in quantitative analysis of the fiber bundles. So the researchers should remain cautious when interpreting the results in research and clinical applications. So thank you. That's the end of my talk. And I would like to thank my lab members and my close collaborators from the O'Donnell lab, the LMI, the SPL, the PNL labs at the Harvard Medical School. And here is my contact information. Please feel free to contact me if you have any questions related to my talk. Thank you.
So yeah, no audience response system for this one, but please feel free to contact Fan. We're going to take a brief break before the next talk, so we'll come back at 10:55, which is in just over 10 minutes, for Marika and for our final set of talks, which is focused on applications of tractography.